What is evolutionary biology? First of all, we shall go through some of the disciplines in biology. Consider physiology. How does the organisms obtain energy from the environment and use it to carry out the life processes? And how does the processes that sustain life are carried out? Physiology is all about addressing this question. Come to biochemistry. What are the chemical activities involved in the so-called physiology? That is what biochemistry deals with. Developmental biology is addressing the question, how do an entire organism develops from a small single cell? Essentially, it is about how the genetic information is translated into observable characters. How does the information is stored and transmitted in organisms? That is what the study of genetics means. When you come to evolutionary biology, almost all of the disciplines are included in it. Evolutionary biology particularly asks this question: why does there are several types of organisms present in this planet and how did they form? In this question, you can see the major component, several types of organisms. This accounts to the biodiversity of the planet, and the study about this diversity include taxonomy. Now, what do you mean by several types of organisms? It means that there are differences between the organisms present on this earth. That is what we call as variations. Each organism is different, and we can distinct and classify organisms based on the variations and similarities. So, now we can see that there are several types of organisms on this earth. We observe them as different types because there are variations between them. And how do these variations occur and how does it formulate a species or organism is what we study in evolution. These variations occur at all levels. There are morphological variations, that is variations in appearance. There are variations in the anatomy, physiology, behavior, etc. Other disciplines of biology study about the mechanisms of these variations, but evolutionary biology basically address the question why and how did these variations occur. Because by understanding these variations, we can come to know why there are different organisms, and different behaviors for all these organisms. Next, we will ask the question why there are variations. What we can see by our naked eyes are morphological variations. Traits, phenotypes, characteristics are all more or less similar terms even though they have a narrow meaning in their specific disciplines. Those variations what we can observe by our naked eye are called morphological traits or characters or phenotypes. We know that genes are responsible for all the characters we have. Our genes encode proteins or enzymes which further catalyze all the biological process. The central dogma of molecular biology states that the information passes from DNA to RNA to protein. Not only the genes encode specific protein, they also regulate the amount of protein produced. Thus, we can understand that the basic reasons for the morphological variations is our genome or DNA. The genome of all life forms, whether it is RNA or DNA, is written in the format of nucleotides. And how does this genetic code is translated into a protein, or how does an information on the genome is converted into a morphological trait is well understood. All these understandings point out one thing: the basis of the morphological variations lie on DNA. Two species are different, or even two individuals are different because they have differences in their genome or DNA. Why there is a variation in DNA? DNA is basically a chemical molecule, and there are 4 different types of nucleotides because of the 4 different types of nitrogen bases present in it. We refer them as A, T, G and C. The combination of this sequence of nucleotides determines the information present on the DNA. Since this is a chemical molecule, there can be changes in the nucleotide sequences, either while replication of the DNA or due to environmental factors. These changes in the genetic sequences are called mutations. Mutations are changes or alterations in the nucleotide sequence. These changes may alter a single nucleotide or a stretch of several nucleotides or even a section of the chromosome. The mutations may arise spontaneously due to errors in replication or DNA repair. Also, environmental factors such as UV light and chemicals can induce mutations. There are two types of mutations based on its transmission. Hereditary mutations are those which will be transmitted to the offsprings. It is because the nucleotide alterations takes place in the genome of reproductive cells. Therefore, such alterations pass on to the progeny. Acquired mutations are those mutations which occur on the somatic cells. These mutations do not pass on to the next generation. Earlier, we had discussed that DNA is the basis of the traits or characteristics. So, if there is a mutation, will it change the trait? Certainly. Mutations result in the change of the morphological characters because the information they have is changed. Here in this example, you can see how does a change in the single nucleotide results in a different protein or a non-functional protein. Based on the phenotypic effect, mutations may be classified into different types. Generally, a mutation may be either beneficial, neutral, or lethal. As the name suggests, a beneficial mutation gives some advantage to the organism. It may be some kind of new function or ability. For example, a mutation which results in change in the smell of a flower, and because of the smell, if more pollinators are arriving, it will be a beneficial mutation. Neutral mutation will not have any effect on the phenotype. Even if it have a phenotypic change, it may not provide any advantage to the organism. Lethal mutations are those mutations which will kill the organism. For example, a mutation can result in a truncated protein that was essential in a biochemical pathway. The organism may die due to the inactive protein. Such mutations are called lethal mutations. Now, think of a situation where a mutation provides reproductive advantage to the organism. Reproductive advantage simply means that the organism can produce more offspring than the others due to the new function gained by the mutation. Or it can also be something which will better support the survival of the offsprings or mate selection. An individual with such favorable mutation will have a better chance to reproduce or raise their offsprings, and they will be naturally selected in the mixed population. Please do not misunderstand that natural selection is something that nature purposefully selects. In a population of individuals with variations in trait, those individuals with a reproductive advantage naturally get selected over the others. These organisms tend to produce more offsprings, and the particular trait will override other traits in the population, and we call it as fixation of the trait. There is one more point to add. The definition of beneficial mutation solely depends on the situation. A mutation that is beneficial for an organism at one habitat may be unfavorable for another. Now we are clear with the simple idea how the biodiversity has come into existence in this planet. Mutations leads to variations in phenotypes, and those variations which are advantageous to the organisms get naturally selected in a population. When mutations accumulate in these organisms to such an extent that two different individuals cannot interbreed anymore, they evolve into different species. In addition to mutations, there is another major factor responsible for genetic variation in sexually reproducing organisms. It is recombination event happening in reproductive cells. Maternal and paternal copies of chromosomes exchange the segments of their genome, which result in a new variant. Also, the independent assortment of non-homologous chromosomes in the metaphase 1 stage of meiosis result in the shuffling of paternal and maternal chromosomes. This also results in the new combination of traits.